dear committee members. Today I'm going to present my thesis defense, which is liquid to soft-solid transition of cellulose nanocrystal suspensions in presence of Micelle-forming surfactants. This is the content of my presentation. In the introduction part, I'm going to talk about my motivation for this thesis, and after that, in the literature review, I'm going to show relevant studies and I'm going to introduce the materials I use in my projects. In the third part, which is project 1, surfactant-driven gelation of cellulose nanocrystal suspensions, I use cationic surfactants to achieve cellulose nanocrystal suspensions gelation. In the second project, which is temperature-induced gelation of cellulose nanocrystal suspensions, I use the temperature-responsive polymer to obtain a temperature-responsive CNC-based hydrogel. And in the final, in the conclusion outlook part, I'm going to have my final comments and I have a proposal. I'm starting with the introduction. And my motivation of the thesis is related to soft matter science because soft matter science deals with complex materials which are in the spectrum from solid to liquid. We cannot classify soft matter as directly liquid or solid. And they have 3 common characteristics. They have structural components at the mesoscopic scale, and the macroscopic behavior is dependent on this relation. They do not have long-range order like crystalline structures have. They have short-range order, and they are more susceptible to temperature changes. And they can undergo deformation under mechanical stress and also electrical and magnetic influences. So it means that soft matter are susceptible to create smart materials. Soft materials are great address. So my aim is to develop non-toxic, environmental friendly, inexpensive, tunable CNC-based hydrogels using surfactants. In my first project, I used 2 identical cationic surfactants. And by using 2 identical cationic surfactants, I just wanted to understand the effect of surfactants counter-ion influence in gelation and their micelles effect in gelation. In the second part of my thesis, I used a non-ionic thermo-responsive surfactant, which is commercially named as pluronic F127, and by mixing CNC and F127, I obtained a thermo-responsive CNC-based hydrogel. So in the literature review part, cellulose is the most abundant polymer in the world, and it is the main component of the plant cells, and it can also be found in bacteria, algae, tunicates, and oomycetes. It's a linear polymer. Its monomer is glucose, and these glucose molecules are bound to each other via 1,4 beta-1,4 glycosidic bond. As you can see that this duo is called a cellulose unit. And the number of cellulose units in complex cellulose molecules is not constant, and because of that we call cellulose polymer as a polydispersed polymer, and degree of polymerization can go up to 15,000, and it is around 3,000 and 4,000 in some other species. It depends on the source of the cellulose degree of polymerization. So we can say that the crystalline order is also changing among species. So there are numerous features of cellulose. They are tough, less dense, and as I mentioned that they are abundant. Since they are coming from nature, they are both biodegradable and biocompatible. And they are water and many organic solvent insoluble. So it makes them suitable for chemical modifications. Cellulose nanocrystals are coming from bulk cellulose. Bulk cellulose has 2 regions, amorphous and crystalline regions. As you can see from this cartoon, if we take a fiber, it is formed by microfibrils, and microfibril structure has both crystalline regions and amorphous regions. Crystalline regions has high order and their mechanical properties are superior. And because of that, we are more interested in these crystalline regions. But these regions are not found in nature isolated, so we need to perform some chemical isolation methods or mechanical isolation methods. High pressure homogenization, enzymatic hydrolysis, and sulfuric acid treatment are one of them. Our CNCs were purchased from Celluforce company, and they are obtained by sulfuric acid treatment. This strong acid gets rid of these amorphous regions. Crystalline regions has superior characteristics. High strength, large surface area. They have extraordinary optical, thermal, and electrical properties. Their Young's modulus is higher than concretes, their tensile modulus is higher than steels, and they have high surface area, which make them suitable for chemical modification and absorption of different molecules. Their length is around 100 nanometers, and their diameter is between 1 to 50 nanometers. Since they are superior, they have superior mechanical properties, mixing nanomaterial with a suitable matrix gives us a new composite material. And there are lots of instances in which the CNC is used as a mechanical reinforcement agent. When CNC is mixed with PVA, PLA, or PO matrices, we obtain a new material which has higher, better mechanical characteristics. It can also give flexural strength when it's used in concretes, and sometimes it is used in coatings. These materials, CNCs, are not able to conduct electricity. However, they can be used in biosensors, capacitors, energy storage devices because they control travel of ions within an electroconductive matrix. And one example of CNC, they can be used in optical films also. Aqueous CNC suspensions, the evaporation-induced self-assembly applied on aqueous CNC suspensions can give us optical films with different properties. But the most important applications of CNC are coming from biomedical applications because they can be used as a drug delivering agent, gene delivering agent, and they can be used in tissue engineering applications as an extracellular mimicking material. And because of that, the development of aqueous CNC suspensions and also CNC-based hydrogels is necessary. And I'm narrowing down my discussion to CNC-based hydrogels. Before going further with CNC-based hydrogels, I would like to have a comment about aqueous CNC suspensions. At low concentrations, these suspensions are isotropic. At intermediate concentrations, their phase behavior is isotropic and liquid crystal. At higher concentrations, they behave like gel. I am more interested in CNC-based hydrogels having aqueous CNC suspensions at the intermediate regime. So these ones are not gel itself, but inducing some chemically cross-linking or physically cross-linking agents, we can create some CNC-based hydrogel. As you can see in this schematic, by introducing some functional polymers, it's possible to create some chemically cross-linked hydrogels. The drawback is hydrogel. The drawback is this type of hydrogel is that they are less responsive because chemical bonds are more strong and they are more stiff. In the case of physically cross-linked ones, simply just addition of salts, polymers, and surfactants are enough to initiate some interaction, some hydrophobic, hydrophilic, and electrostatic interaction. And by means of these interactions, physically cross-linked gels can be obtained. And there is an advantage of these gels. They are responsive. We can say that they are smart. So before going further with my CNC surfactant mixtures, briefly I would like to talk about surfactant. I am taking this cationic surfactant group. Surfactant has a hydrophilic side and also hydrophobic side. This resembles polymer and this resembles salt. If the surfactant concentration in aqueous suspension is high enough, we observe some micelle formation after the critical micelle concentration. So in our discussion, CNC surfactant mixtures, we need to classify the type of surfactant. If the surfactant is anionic, we observe hydrophobic to hydrophobic attraction because the negative charge does not want to close to negative CNC groups. And the other type of interaction between them is via a positive counter-ion bridging. We observe gelation mostly in cationic surfactants because there is a great interaction between the positive cationic group and the negative CNC groups. By means of these hydrophobic attractions between the tail groups, we observe the gelation. In nanoionic surfactant, the type of interaction depends on the type of the nanoionic surfactant. If this is, it can be either hydrophilic to hydrophilic or hydrophobic to hydrophilic type interaction. In Zwitter ionic surfactants are surfactants having both plus and minus group in their heads. The type of interaction is similar to cationic surfactant interaction. So I'm continuing with my first project, which is surfactant-driven gelation of cellulose nanocrystal suspensions. My aim my aim is to develop a CNC-based physical hydrogel by using two similar cationic surfactants. As I mentioned that cationic surfactants interact with the negatively charged CNC group and they induce gelation. However, in my study, I would like to understand the difference caused by the counterion. As you can see that both surfactants having the same head group and tail group, the only difference is their counterions. 1-decyl-3-methyl imidazolium chloride and 1-decyl-3-methyl imidazolium ferric chloride are my surfactants. There is one interesting fact about this surfactant because this surfactant is also a magnetic ionic liquid, as you can see in this picture. This surfactant is susceptible to magnetic field. So by means of this study, I would like to understand the effect of counterion in gelation and also this surfactant for micelles at similar critical micelle concentrations. However, their micelles electrochemistry are different. And because of that, I am going to also understand the effect of charge of micelles in gelation. So at the final, I will understand the final gel's mechanical properties. So I started my experiment with electrical conductivity measurements to understand, to see the critical micelle concentration of both surfactants. As you can see here that the electrical conductivity is linearly related to surfactant concentration. And at the point where the trend leaves the linearity is noted as the critical micelle concentration. For chlorine-containing surfactant, the critical micelle concentration is around 60 millimolar. For iron-containing one, it is around 46 millimolar. We can say that the critical micelle concentrations are not identical but similar. In the dynamic light scattering measurements, I just wanted to understand the size of these micelles. I prepared 100 millimolar solutions and I just made an experiment, dynamic light scattering experiment, to see their sizes. As you can see from this size distribution, chlorine-containing one has 1.5 nanometer size and the other one is 1.89 nanometer respectively. This shows that micelle size and shapes are similar. But the only difference between these two surfactants, the micelles formed by these surfactants have different zeta potentials. Chlorine-containing one have a positive zeta potential, whereas the other one is negative. And CNC is inherently negative, has a negative potential because of the sulfate ester groups on the surface. So in another experiment, in small angle neutron scattering measurements, I just wanted to understand the micelle-micelle interactions. These experiments were conducted in the United States in National Institute of Science and Technology. So as you can see that the intensity profiles were taken for below, above, and well above critical micelle concentration because our critical micelle concentration is around 60 millimolar. As you can see that intensity is the result of the form factor times structure factor. When we look at that, below 70% for this surfactant type, we do not observe any critical micelle formation, and we also do not observe micelle formation for this surfactant below 60 millimolar percent. And the other thing, structure peak factor, we can mention structure factor when there is a peak in the intensity profile. So we can see that there is a peak, it shows that there is a structure factor. Structure factor means that there is an intermicellar interaction, there is a Coulombic repulsion between two micelles. So our SANS measurements also give us an idea about the intermicellar interaction between two micelles. And the shape of these micelles, according to our fitting by using a certain software, are prolate shape, and their sizes are also in great agreement with our DLS measurements. After having a comparison in nanosize, I just wanted to understand the ability of surfactants turn CNC solutions into gel. So because of that, I conducted a tube inversion test for 1% CNC, 2% CNC, 3 and 4% CNC containing complexes. And I converted these images as a sol-gel transition map. as you can see that these two maps, this one showing the chlorine, this one showing the iron containing one, always black and red, and they are similar. However, there is a slight difference in this region, and this slight difference, which happens at 2% CNC content and 10 and 50 millimolar surfactant concentration, this is attributed to the hydrophobic nature of the ferric tetrachloride counterion. Because it's hydrophobic, it does not want to disperse in water, it stays with the surfactant group. Therefore, the number of surfactants attached on the CNC surface are not too much in this case. For example, in this case, since chlorine can well in water, and because of that, there are much more surfactant groups adsorbed on the surface. So by means of this hydrophobic attraction between the surfactant groups, we obtain gels. So I would like to have a qualitative analysis for the gel samples because this analysis was not enough. And because of that, I wanted to perform a rheology analysis. Rheology is interested in soft matter and is the science of flow and deformation. So the application time is also important and magnitude of stress or strain is also important. The soft material, for this case, 3 or 4% CNC containing gels were placed in between two parallel plates. This one is stationary in raw matter. This one is rotating. This is rotating and this is rotating with a this applies an oscillatory strain, as you can see from this formula, sinusoidal strain. So the response of this material is the also sinusoidal oscillatory stress. This is recorded by the rheometer. However, this has two components as you can understand from this shift angle. So one response is real and the other response is imaginary. As the modulus is the ratio of stress over strain, you can easily see that modulus is dependent on time because these two functions are dependent on time, and modulus has two components. The first component, which is coming from the in phase with applied strain, elastic response. The other one is out of phase with applied strain, viscous response. This shows the solid-like behavior and this shows the liquid-like behavior of the matter. And also, there are two kinds of rheology analysis. As you can see in frequency sweep experiments, the strain changes, the strain does not change with the time. It shows that the only frequency changes. It is frequency sweep experiment and it gives us information about the time-dependent behavior of the material. In amplitude sweep experiments, a constant frequency is selected. However, the amplitude increases. So it gives idea about the behavior of the material under changing deformation at a constant frequency. So my first test was frequency sweep test. I use frequency sweep test to understand whether these samples are actually gel or not. As you can see from these figures, both solid, the filled ones showing the storage modulus, the other ones showing the loss modulus, the response of these materials are independent from increasing frequency. This is a great example that these materials are itself gel. However, when we look at the neat CNC at 3%, it is not independent of frequency. This is the great example that this is a viscoelastic liquid. So I had the frequency sweep experiment in linear viscoelastic region and I applied strain values between 0.1 and 1% between this frequency range. This is the also a SEM image of my gels. You can see the meshes. In amplitude sweep experiments, as I mentioned, at the constant frequency, strain is increased from 0.01% to 100%. So it is not only limited to linear viscoelastic region, we also observe the nonlinear viscoelastic region. By means of this analysis, we can have an idea about the microstructure of the gels. According to Wynne et al., there are three types of complex fluids, as you can see. The first type is strain thinning, strain hardening, type 3 weak strain overshoot, and type 4 strong strain overshoot. But we always observe type 3 behavior in our complexes because this belongs to polymeric mixtures. And since CNC is a polymer, we observe the polymeric mixture behavior. And I would like to introduce the important points in this schematic. As you can see that storage modulus starts at the G plateau value. This is the highest value. At this level, gel is intact. But after application of some small strain, we start to observe some micro scratches. And because of that, gel, the application of the strain is no longer in the linear viscoelastic region. After a certain point, we realize that gel is not intact anymore and flow takes place. The increase in the loss modulus also the result of the energy loss during the breakage of the crosslinks. This flow point is also noted as critical strain point. And my amplitude sweep, these are the results from my amplitude sweep experiments for 4% CNC containing ones at 10 millimolar surfactant concentration, black one showing chlorine and the other one showing the iron, 50, 100 millimolar, 150 and 200 millimolar. As you can see that these are well above critical micelle concentration. At this concentration, we are observing some micelles. Here, we do not observe some micelles. If I start the discussion from chlorine containing one, as you can see that when the surfactant concentration is increased, we observe that also storage modulus increased. The increase in storage modulus means that number of crosslinks increased. So this is related to adsorption of the surfactant groups on the CNC surface. Further addition caused formation of the micelles, but micelles are also micelles are also positively charged and they can also have a great interaction with CNC, but some surfactant groups leave the CNC surface, and because of that, we are observing some small decrease. At this point, and this is 10 to the 5. And after that, we observe that there is slight decrease and there is slight decrease again because of the formation of free micelles. These free micelles affects the other micelles which are serving as a cross-linking point. For this case, actually, since I mentioned that ferric tetrachloride is hydrophobic and it just wants to stay with the surfactant group, and because of that, at this surfactant, increasing the surfactant amount does not mean anything about changing the mechanical property. As you can see that at this concentration, micelle formation is occurring also for iron containing one, but the effect of micelles, since these micelles are neutral, is not really drastic. So I just wanted to summarize my amplitude sweep findings in this page. As you can see that a chlorine containing one's storage modulus increase when the surfactant addition, when the extra surfactants added the system. But after the critical micelle concentration, these number of crosslinks also reduce. This is valid for this case also when we increase the CNC concentration. However, we cannot observe the same effect in the iron containing one. And this is a graph showing the critical strain. As you can see that the higher the critical strain means that the material is less brittle. It means that the dominant interaction type is not electrostatic. However, when we go further, the critical micelle concentration, we observe that this material, chlorine containing one, becomes more brittle for the two cases. For these two cases, it shows that these micelles are also positively charged, and these micelles, this is showing this, these micelles are not electrostatically attracted to the CNC. So my findings are published in a journal LANGMUIR with the name of surfactant-driven liquid to soft-solid transition of cellulose nanocrystal suspensions, and I had a proposed mechanism as you can see here. Below critical micelle concentration, we do not observe any micellization. The only attraction is between this hydrophobic attraction between the tails. Above critical micelle concentration, we observe electrostatic attraction of these micelles to the CNC surface, but we only observe hydrophilic attraction of these micelles on the surface because they are neutral micelles. Well over critical micelle concentration, there are more free micelles, and the more free micelles has a long-range electric repulsion for the micelles behaving like a cross-linking point. But we only observe short-range repulsion in these samples. This is the explanation why these samples are more brittle than this one. This is the first time a comparative study about the effect of micelles in literature, and we just wanted to make a comparative study to show the effect of micelles. In the second project, which is temperature-induced gelation of cellulose nanocrystal suspensions, a CNC-based injectable and thermo-responsive hydrogel was wanted to be developed by using a non-ionic surfactant. This non-ionic surfactant, as you can see that, this has ethylene oxide, and propylene oxide side. It is it's a block copolymer at the same time, and its commercial name is also pluronic F127. This polymer itself is thermoresponsive, but there is no study in the literature about mixing these two and obtaining a new CNC-based hydrogel. The aim of this project is to develop some material which is able to dual drug delivery, because both of these components, both pluronic F127 and CNC, are able to convey drug, and dual drug delivery systems are promising, especially in cancer treatments. So by means of this study, we would like to understand the CNC's effects on phase transition temperature, because we are adding CNC to these aqueous suspensions, thermoresponsiveness and mechanical recovery, because it itself mechanical recovery, it has a mechanical recovery. And we would like to understand the effect of CNCs on mechanical properties of the final complex. So at first, again, we had the dynamic light scattering measurements. As you can see that these form at low temperatures, these form unimers. At higher temperatures, it forms micelles. And by the increase of temperature, we observe formation of micelles, and at higher temperatures, these micelles start to interact by a hydrophilic attraction, hydrogen bonding, and form an array of micelles. So at first, we showed that neat CNC solutions are not responsive to temperature increase. To show the temperature responsiveness of these materials, we had a viscosity versus temperature measurements, and we use steady shear rheology analysis, and the steady shear was 1 Pascal, the steady stress 1 Pascal. And as you can see that neat CNC solutions are not responsive to the temperature in between 10 and 50 degrees. The only difference is caused by the addition of different increase in the CNC concentration, because when CNC concentration increases, viscosity also increases. In the second graph, neat F120 solutions, we observe that there are gelation for these 15, 17.5, and 20% F127 containing ones. But we did not observe any gelation for this regime. However, you can see the slight increase in here. This is the proof that there is a micellization. But this micellization was not enough to make a complete gelation. So after that, we combined CNC and pluronic F127 mixtures. And as you can see in this graph, this has 12.5% F127, and we already know that neat form of pluronic is not responsive, does not undergo gelation. However, as you can see that for 1, 2, 3%, 4%, and 5% CNC containing ones, there is no sudden gelation also observed for this concentration. At 15% pluronic F127 containing ones, neat form can undergo sudden sol-gel transition, but only 1% CNC containing one undergoes sudden gelation. Sudden gelation is important for injectability also. In 17.5% pluronic F127, as you can see that neat form 1, 2, 3% CNC containing ones undergo sudden gelation. At the last form, actually all of them are solid, all of them are gel, but since there is no sudden gelation for these cases, we are not interested in them. We are not interested in them at all. And the same trend is observed for 20% plurionic F127 containing ones also. Need 1%, 2%, and 3% containing CNC containing ones undergo sudden gelation. So I just wanted to make sure that the gelation, CNC concentration is affecting the gelation. As you can see that at 20% plurionic F127, increasing the CNC concentration inhibits the gelation because the gelation temperature increases further. And this is valid for 17.5 case also. We observed the same trend. However, there is not the same trend for 15% containing one, and we attributed this difference to that since this is an intermediate region, and we were not able to interpret it very well. These are the images, tube inversion tests, as you can see that. At 10 centigrade, there are liquid. When they are increased to the body temperature, because we would like to use them as a drug calibratory systems, we observed that all of them are gels. So another aspect of CNC, we would like to understand their effect in thermo-reversibility. How did we compare that? We just plotted the viscosity versus temperature graph, and we recorded the heating path. As you can see that the black ones showing the heating path for the neat one and the empty ones showing the cooling path. As you can see that the final, the beginning of the viscosity and the final viscosity is equal to each other. Even though increasing the CNC concentration, as you can see here, 3%, 4%, and 5%, did not induce any negative effect in thermo-reversibility. All of them are thermo-reversible. So again, in oscillatory shear analysis, it is important to understand the microstructure mechanical features of the gel. So my application strain in frequency sweep experiment was 1%, and it is only performed for 17.5% plurionic ones because all of them are gel at this temperature. And this is the frequency versus modulus graph. And as you can see that solid storage modulus is always higher than loss modulus. This proves that the resultant materials are gel. And in the phase angle section, and also you can see that the most phase angle is 15, and if the phase angle is less than 45, it means that the resultant material is gel. So another thing about CNC, I just wanted to understand CNC's effect on the mechanical properties. As you can see that storage modulus, higher storage modulus means more cross-link points. In the neat form, always high cross-link points. However, when the CNC concentration increase at 4% and 5%, we observed that they have similar amount of cross-link points. So I also made a plot about the storage modulus and critical strain in which the flow occurs and gel is not intact anymore. And the reason of having the number of highest crosslinks at this point is that neat F127 micelles interact with each other and they form a ordered array. And addition of CNC just eliminates that, is not in favor of this array, and because of that we observe that number of crosslinks reduces. For the critical strain thing, in lower critical strain means that this one is the brittle, has a brittle structure, but increasing the CNC concentration make them less brittle, because at higher CNC concentration, the main component of the gel becomes CNC, even though the main component of the gel was F127 at lower CNC concentrations. I can basically say that CNC and F127s are two competing agents to make the forms. At lower concentrations, F127 is the main component, and at high CNC concentrations, they become the main component of the gel. So also, these are the summary of my amplitude sweep experiments. High storage modulus means high number of crosslinks. The highest number of crosslinks are in neat sample. because of this structure. My cells are forming a ordered array, and under low shear, they can stick off my cells and slip off layers motion is seen. However, when we add the CNC, we do not observe the same behavior, and because of that, they are critical, and this is proven by their increase of the critical strain. So the last test is their mechanical recovery test. Since I wanted to have an injectable CNC-based thermoresponsible hydrogel, it is necessary to understand their mechanical recoverability. So under low strain, certainly the storage modulus is higher than the loss modulus. When the strain is applied, the gel is liquefied, as you can see that. Loss modulus is higher than storage modulus. But when we withdraw this strain, we observe that the resultant material is also gel because it has a higher storage modulus than loss modulus. These experiments were performed for these samples, and this is a great example that the developed material can be used as an injectable hydrogel. So my conclusion is that CNC addition increases the gelation temperature. It's not in favor of gelation. However, if you would like to use CNC as a drug conveying agent, it is necessary. Structure become less ordered when the CNC is added to the structure, as you can see here. And final complex can undergo thermal and mechanical recovery because we performed that thermo-reversibility is available and mechanical reversibility available for these two gels. And this is a great potential for dual drug delivery purposes. This is my explanation for the mechanism. At low temperatures, there is no micelle formation, and as you can see at concentrated CNC concentration, the gelation does not have a huge impact because it's already close to the gel structure. The gelation micellization of F127 does not have a huge impact. So I'm going to my final comments in the conclusions and outlook section. So in the first project, we saw that in order to tune gel stiffness, micelle charge and also the counter-ion of the surfactant is effective. And also, it doesn't mean that addition of high surfactant doesn't mean that high number of crosslinks. So there is no linear relation between them. So in the second project, we were able to develop CNC plurionic F127 mixture complexes, which are thermally and mechanically reversible. And by means of these mixtures, dual drug release systems can be developed. And this is my suggestion for the second project. When we have the solution phase, both plurionic F127 and CNC, we can conjugate some drugs via UV cleavable bonds. And after the heating, we can inject this material to the human body, for instance. These become gel and it can be stayed in the human body. And by means of light, since these bonds are UV cleavable, we can achieve the control